Sure. Sure.
Okay, good evening, ladies and gentlemen. Rather, good afternoon in the UK. I'm sure it's evening where a lot of you are positioned. Welcome once again to another session of Thoracic Gurus at AZK. Today we are going to talk of two very, very important topics. The first one is something that you will actually not come across textbooks. Certainly not in surgical textbooks, and we are very privileged to have with us Professor Akif Turna. Professor Turna, as you know, is a professor of thoracic surgery at Cerrahpaşa University in Istanbul, and he's a very well-known gentleman who has published extensively on lung cancer and particularly in terms of mediastinal lymphadenopathy. And he has got a large series of probably one of the biggest series across the globe. And Professor Turna is going to talk to us today about tumor immunology. So may I request everybody to please switch off your microphones? That's the first thing, so that we can listen to Professor Turna uninterrupted. Welcome, Akif. Please share your screen. There's a green button at the bottom. And carry on. Thank you very
everyone.
much.
Thank you very much for this very kind invitation again. So I'm going to talk about this subject. I'm very, I feel very lucky to talk about this subject because I love immunology, and I have been working about this subject for at least 25 years, and we have been conducting some of the experiments and some studies on this subject, but it is actually not a common subject to talk about, but during the last 5 years it has become very important because some of the very effective anti-tumor immunotherapies have become some kind of mainstream therapeutical option. And today, as thoracic surgeons, actually we are obliged to know some main mechanisms of anti-tumor immunity. So I'm going to talk about carcinogenesis first with a couple of slides, because in order to understand anti-tumor immunity, it is very important to understand the mechanism of carcinogenesis and the proceedings that are involved with carcinogenesis. As you know, carcinogenesis involves two main molecular mechanisms. One is oncogene activation, the other one is anti-oncogene suppressions. So oncogene activations include many oncogenes, and these activations sometimes are rearrangements, some are done by rearrangement of the genes. It happens in MYC, ERK1 and other oncogenes, and mutations are important in order to activate oncogenes. For example, in KRAS oncogene activation, it takes a mutation, very specific mutation, and it is true for a couple of other mutations, other oncogenes. Actually, there are approximately 100 oncogenes that are involved with carcinogenesis. And also, some of the anti-oncogenes should be suppressed or should be mutated. Anti-oncogenes are some genes that involve with some very important cellular mechanisms, including repairing of the gene and controlling the cell cycle mechanisms. But if some of these anti-oncogenes are mutated or are deleted, or if there is a deletion in some of the parts of the oncogenes, they become activated, and they become one of the molecular mechanisms of carcinogenesis. And there are some epigenetic mechanisms. As you know, one gene in some situations does not lead one protein. After transcription occurred at nucleus, there are some epigenetic mechanisms that occur just after transcription. And for this reason, even the fact that one DNA is transcribed into RNA, the RNA can be changed by some epigenetic mechanisms that lead to many different proteins or parts of proteins, which are peptides. So from normal cell to tumor, it doesn't happen at one step. There are some multiple steps that should be completed in order to reach tumor cell. As you know, there are two dysregularities about a cancer cell. A cancer cell loses the proliferation control, and also there is a problem with differentiation. It means that the tumor cells proliferate indefinitely like bacteria, and these cells usually do not do their jobs, which can be defined as indifferentiation or dysregulation of the differentiation. So, but as I told you, it doesn't happen at one step. It doesn't happen once. A normal cell, because of some mutagenic effects, actually becomes a hyperplastic cell, and this process is reversible, and dysplasia can occur after another mutagenic effect. It takes more than one mutagenic effect or mutagenic actions for the cell to become a tumor cell, a cancer cell. Even these tumor cells cannot metastasize without some specific additional mutations that can lead to metastatic potential for neoplastic cell. So also, besides the tumor genes, oncogenes and anti-oncogenes, there is a dysregulation about apoptosis. Apoptosis regulates the timely death of cells. Cells should undergo death when there is a time for death. Actually, it is like a indispensable fate for the cells. But the apoptosis of cells can occur by two main mechanisms. One mechanism involves caspase-9. The other mechanism involves caspase-8. And as you see here, this mechanism, this pathway, involves some of the compounds, some of the molecules that are important for immune system also, such as TNF. As I showed you before, a normal cell cannot be a tumor cell at once. For example, it has been very nicely identified that a glial cell can be astrocytoma with some of the specific gene dearrangements, as you see here. And astrocytoma can become anaplastic astrocytoma with the addition of some other mutations. And lastly, a glioblastoma can occur, it can be developed after some of the additional other mutations at some of the specific genes. So these mutations, these dearrangements or deletions should occur for the genesis of glioblastoma. This is a good news and a bad news. Good news, it is a good news because these genes can be repaired at some steps. This is why tumor cells even the fact that the mutation is not so uncommon, not so infrequent, tumor cells can occur very frequently. But the bad thing is that some of the steps cannot be reversible, and these cells can proliferate, and in these cells, the mutation rate is very high than the normal. So, of course, in terms of lung cancer, some of the oncogenes that are important for carcinogenesis have been very well defined. KRAS, BRAF, HER2, MET mutations are the most important ones. P53 can be found in approximately 50% of the 50% of adenocarcinoma cells, and even it can be higher. It can be found higher at a higher frequency in squamous cell carcinoma, and approximately 3/4 of cells have of the small cell lung cancer cells have actually P53 mutations. So these mutations can be identified on the tumor cells on the tumor cells, but we cannot know which mutation leads to which tumor cell. So DNA damage is important, but these DNA damages should lead to many dysregulations about the cell molecular mechanisms of the cell. There should be cell cycle arrest. There should be caspase activation, which is related to apoptosis. Premature senescence is one of the features of the tumor cell, and also proliferative senescence is another characteristics of the tumor cell. So one mutagenic event can lead to many dysregulations in one cell. There are also some of the other differences in terms of expression. Actually, in tumor cells, even the fact that some of the genes are not mutated, are not changed, the expression can change, the amount of expression can change. For example, E-cadherin and WNT are important molecules for the interaction of the cells with the intercellular system, the stroma. The expression of these molecules change. Usually, it is decreased. Actually, this decrease leads to very loose interaction with the extracellular matrix, and also this decreased expression is one of the reasons that these cells, these tumor cells, can metastasize very easily. So there are some genes and proteins changed, but also some of the normal proteins expressions change in the tumor cells. But there is one fact that that seems a good news at first sight. Whole tumor mass actually comes from only one cell, which is called as a monoclonal theory in tumorigenesis. But the problem is that this tumor cell can change very easily. The mutation rate is about 100,000 times higher than the normal cells. Actually, it varies between 10,000 and 1 million times. It means that after the genesis, after the occurrence of one tumor cell, tumor mass comprises many different cells because of the high mutation rate. So it becomes more and more difficult to identify these tumor cells in terms of expression or mutations of the oncogenes. So we come across with a very different sets of tumor cells when we see these tumors clinically. Chronic inflammation is one of the important things about carcinogenesis. Nitric oxide and oxygen radicals are the molecules that can transform the normal cell. So can we say something about the clinical picture of the tumor when we look at the carcinogenesis? The answer is yes and no. Yes, because some of the inflammatory factors such as tuberculosis usually lead to some specific cell types such as adenocarcinoma that tends to be more peripheral, as you see here. But tobacco smoking actually cause another sets of mutations. Usually it involves with the p53 mutations, and these factors actually lead to another types of cells that are usually squamous cell carcinoma and small cell carcinoma, and these carcinomas are diagnosed usually at the more higher locations. So this step I aim to reach the anti-tumor immunity. But as one of the latest slides about carcinogenesis, tumor environment, I would like to say that tumor environment is one of the most important things, because these tumor cells, when they become a mass, very small mass, it creates an environment including some of the inflammatory mediators, some of the molecules around the tumor cells, and also it creates a chronic inflammation. And this inflammation and these new factors, the mutations and the changed molecules, recruit to macrophages and neutrophils first. This is why I would like to talk about anti-tumor immunity in a chronological sequence, because at first sight, in the front line, neutrophils and macrophages begin to attack the tumor cells, because they are components of innate immunity and they don't need specific receptors to recognize the tumor cells. As I told you, neutrophils and the macrophages are the first cells to approach the tumor cells, to attack the tumor cells. And also polymorphonuclear myeloid-derived suppressor cells come to the site, the front line, not for the activation or attack the tumor cells, but to suppress the anti-tumor immunity. So it is fair to say that at the beginning of the anti-tumor immunity, there are some cells that produce anti-tumor cytokines and the molecules that stop the tumor cells. Actually, from one perspective, it is understandable, because tumor cells are some of the changed mammalian normal cells. So in every immune mechanisms, there is one site that attacks, that is responsible for the attacking to cells, and there is one other site. There are some populations of cells that suppress the attack. A typical macrophage actually has many receptors, and some of the receptors, even the fact that none of them are specific to antigen, some of the receptors can bound to the tumor cells. Among them, CD64 is important. If there is an antibody bound to the antigen, which is on the surface of the tumor cell, FC receptor is activated by bounding to the immunoglobulin. And there are some scavenger receptors that can bound to the tumor cells. Actually, mannose receptor is important. Actually, mannose receptor is here because usually it leads to binding to some bacterial antigens. In mammalian cells, most of the carbohydrates are 6-carbon carbohydrates. But on bacteria and some of the other cells that are not mammalian cells have some 5-carbon carbohydrates. So mannose receptors are here because of this recognition, possible recognition. But these receptors can bind to some molecules that are onto the tumor cells. And also CD14 is important in terms of the tumor attacking or binding to the tumor. And some toll-like receptors are important. They work as a toll system. Actually, there is no specific binding site to this receptor, but some of the tumor antigens can bind to this receptor. So what about after macrophage? Macrophage, as soon as they are activated, they become 2 subpopulations. The one subpopulation is called pro-inflammatory macrophage. The other population is anti-inflammatory or pro-tumor macrophages. And these macrophages are suppressive macrophages. Some of the cytokines are secreted to suppress the activating macrophages. And also some of these macrophages can stimulate the effector cells to develop a specific receptors against tumor cells, and other macrophages actually inhibit the T cells. So from the beginning of the activation, 2 subpopulations of macrophages are developed, but unfortunately one subpopulation is developed to suppress the anti-tumor immunity, which is a problem for the organism, of course. So this shows the mechanism of macrophage-mediated suppression. As you see here, there are many pathways to suppress the immune system, immune mechanisms, and these suppression involves with signaling some of the amino acids like L-arginine. It involves also cell-to-cell interactions mediated by HLA-G and HLA-E, and it also involves with some of the apoptosis-inducing molecules, which is called PDL1, programmed death ligand, and also it involves with some of the suppressor molecules such as B7-1. These molecules inhibits T cells, natural killer T cells, and B cells, and also these molecules induce apoptosis, cell death, to these cells. Tumor-associated suppressor macrophages don't only suppress the anti-tumor immunity, they also weirdly and interestingly help the tumor cells them to metastasize more easily. They cover the tumor cells, as you see here. They help to move the tumor cells inside the vascular system, and also they cover the tumor cells in order to protect them from the T cells and the other cells inside the bloodstream. So they actually very effectively help the tumor cells to become more carcinogenic and metastatic. What about the other chronologically the other cells that come to the tumor site, tumor macroenvironment after the macrophages? Natural killer cells are the second wave of cells actually that come to the tumor site, tumor macroenvironment. Normally, natural killer cells have no antigen-specific receptors, but they use very clever system. They actually tend to activate when the adhesion molecule is is bound to the adhesion molecule is bound with an activation molecule, but it takes an inhibitory signal from a normal cell to stop the natural killer cells to be activated. So in normal cells, they don't activate because they recognize that these cells are normal cells. It is a very clever system because they don't need an antigen-specific receptor, but they need an inhibitory signal. Normally, natural killer cells are expected to recognize the cancer cells because cancer cells tend not to express MHC class 1, which is an inhibitory signal. But some of the cancer cells can express MHC class 1 in order to inhibit natural killer cells. Also, natural killer cells are activated by the IgG-Fc component because there is a receptor called CD16, and they are activated against this molecule. So, of course, there is no two molecules. Actually, there are more than two molecules on the natural killer cells, which are related to inhibition and activation. NKG2D and NKG2A molecules are inhibitory molecules, which recognize the HLA and the inhibitory molecules. So what is the killing mechanism of natural killer cells and also T lymphocytes? Actually, they pump water. Simply, they pump water inside the cell. So this shows the mechanism of action of the killing and of natural killer cells. They pump actually sodium inside the cell, and since there is a covalent bond between the sodium and the H2O molecules, the water molecules, there is a water influx inside the cell by the aid of this pumping molecule, which is called perforin. And this perforin is activated by calcium. So T lymphocytes are the most important cells in terms of tumor immunity, and these lymphocytes are seen in every side of solid tumors, and some actually they are active against some of the non-solid tumors like lymphomas or leukemias. They are very active, and they have specific receptors to the changed molecules. And also they work as a conductor. They work as a director of the other tumor cells. They decide what to attack, what to suppress. So this is why the modification of these lymphocytes are so important in order to enhance the anti-tumor immunity. But in order to understand the anti-tumor immunity driven by T lymphocyte, one should look at the antigens that are on the tumor cells. I talked about the mechanism that lead to different type of antigens on the tumor cell. This is why we can see many different type of antigens onto the tumor cells. There are some silent antigens, which means that the expression is too low in some conditions. When there is an attack led by T lymphocytes, some of the antigens become silent. There are some mutant antigens. There are some antigens that are effective in immunological escape, and some of the antigens like MHC class 1 are lacking as a result of the carcinogenesis, and some of the carbohydrate components are altered on the tumor cells, and there are some metastatic molecules. T lymphocytes basically recognize the different molecule, different peptide. So it would be very nice if there is a completely different molecule which is expressed on the tumor cell, but there is no specific molecule to the tumor cell. This is why the recognition of these antigens are very difficult task, because all these antigens are present on our normal cells. So T lymphocytes should find a way to attack these tumor cells. Having said that, some of the molecules are somewhat specific to the some tumor cells, which are which are shown here. Some of the molecules like Gauge family and MAGE3, these molecules are found in some of the testicle areas. As you know, most of the testicle tissues are immunologically privileged size, which means that these antigens are not met by T lymphocytes in normal situations. This means that these molecules can be recognized by very strange molecules, different molecules. But other molecules are usually found in normal situations. There are some viral proteins that can be found on the tumor cells, and there are some changed proteins on the tumor cells. But as I told you, none of the molecules, none of the antigens are specific to the tumor. As soon as the T cell is activated by binding of the cells, the molecules, sorry, these cells are divided into basically three components, three subpopulations: Th1, namely Th1 cells, Th2 cells, and Th17 cells. Actually, Th1 cells are activated molecules like first responder soldiers, and Th2 cells are other component, other population. Usually they secrete some of the cytokines to suppress the Th1 cells, and they are responsible for the humoral immunity. They are responsible for B cell immunity and allergy. Th17 cells are induced usually because of tissue inflammation, since most of the tumorigenesis involves with the tissue inflammation. For example, lung cancer, colonic cancer, and some skin cancers are related to tissue inflammation. Th17 cells are developed, and all these cells are found in the tumor macroenvironment. What do? They do different tasks, actually. Some of the cells attack the tumor cells, and some of the other cells actually help the tumor cells, as you see here. There are more than three subpopulations. Those subpopulations that I showed you are certain subpopulations, but there are other subpopulations of T lymphocytes, such as gamma delta T lymphocytes, MAGE cells, mucosa-associated invariant T cells, which are found in the respiratory mucosa. Th2 cells are here. They secrete some of the cytokines that are suppressed T lymphocytes, and they help tumor cells. So it seems strange, and these cells actually are expected to attack tumor cells, but some of them are helpers to tumor cells. Why? Because tumor cells are usually, basically, fundamentally, are our cells, changed our cells. This is why there is a suppression against our cells. So interaction with T lymphocytes is important, and this interaction actually is caused by dendritic cells, which are antigen presenting cells. These cells are supposed to induce T cells, but some of the dendritic cells suppress T lymphocytes, and they induce tolerance instead of trigger or attacking to tumor cells. So this is why it is a complicated mechanism. There is two actually mechanisms that are against each other. So heterogeneous mechanisms involve many other molecules and the cell-to-cell interactions with ICAM1, PDL1, Fas ligand, and in indole-deoxygenase molecule. And tumor cells create tryptophan depletion, and also that in turn leads to T cell inhibition. T lymphocytes actually should be activator for macrophages, but also T lymphocytes become activator of tumor-associated macrophages, and also indirectly some of the T cells become helper of tumor cells, helper of metastasis, as you see here. So there is a positive feedback between macrophages and the T lymphocytes. Macrophages are the first responders. They activate T lymphocytes, and also tumor activates T lymphocytes, but also these activations leads to activation of tumor-associated macrophages. This increases suppression extensively. So tumor, with the help of T lymphocytes, with the help of the cytokines and the mediators secreted by itself, and also by the help of other cells like tumor-associated macrophages, become a suppressor, become the source of suppression in the tumor microenvironment. So the tumor cells actively become suppressor of the anti-tumor immunity. They protect themselves. As a result of that, T cells can respond and can develop anti-tumor receptors called T cell receptor, and they can bind to the molecules that are expressed by tumor cells, but they can become allergic to tumor antigens, to tumor cells. This is why it is, of course, important to generate an anti-tumor response in order to kill the tumor cells, but also it is important to suppress the allergic T cells in order to increase the anti-tumor activity. So there is no actually a problem of developing anti-tumor immunity, but there is a problem with the process of this anti-tumor immunity, because this anti-tumor immunity is very effectively suppressed by the T cells. And the cytokines should not be forgotten, because some of the cytokines, like hormones of the immune system, actively suppress the effector cells, as you see here. Among them, TGF-beta, interleukin-10, interleukin-23, and VEGF are the most important ones. TGF actually betrays immunity in terms of anti-tumor immunity. There are many cheating and the treason in immune system, and TGF-beta probably is the leader of the treason, if you will. TGF directly inhibits the anti-tumor immunity. So the mechanism is more complex than it should be, because it involves many suppressor and the activation mechanisms. And also there are some intracytoplasmic molecules that are important in terms of the activation of the genes. STAT3 is one of them. Tumor, inside the tumor, STAT3 is activated. STAT3 is one of the activator of some array of the genes. These genes are effective in suppressing the activity. This is why STAT3 is induced in natural killer cells, T cells, and some of the other cells, such as granulocytes. And tumor not only suppresses the anti-tumor immunity by cytokines and cell-to-cell interactions, but also indirectly inhibits the anti-tumor immunity by the increasing of the suppressor molecules, intracytoplasmic molecules like STAT3. So what happens with these suppression and the activation mechanisms? There are three possibilities when there is an anti-tumor immunity. One possibility is that the tumors can activate, and they can be very effective against tumor cells, and they can eradicate the tumor mass. In this situation, there should be no clinically diagnosed or prominent tumor mass. So it is elimination of the tumor cells. Actually, it happens in our system all the time. This is called immune surveillance. Immune surveillance was proposed approximately 70 years ago, and it explains how we eliminate the tumor cells in most of the situations, because mutations are so frequent that theoretically in our system we have approximately five tumor cells that develop every day. But fortunately, this mechanism works. But if there is a suppression that I showed you against activation, there can be equilibrium between the tumor cells and the inflammatory cells. In this situation, we should expect stable tumors that are stable, usually tumor cells that are stable for 2 years, 3 years, 4 years. But if the tumor cells suppression prevails, the cancer cells escape from the immunity, and they become a tumor mass with the aid of chronic inflammation. Actually, Paula Matzinger, this is the picture of her. Paula Matzinger proposed a theory that is very, very important in tumor immunology. In immunology, actually, she is like an Einstein of immunology, because in 1990s, this battle between the anti-tumor immunity and the suppressive immunity could not be explained. But she proposed a concept that is called danger signal. So in this concept, tissue damage can cause a damage signal, and this damage signal can lead to immune response, and it can lead to eradication. Also, by the mechanism that I showed you, it can lead to immune escape, and it can lead to equilibrium. But there is a very delicate balance between immune escape and the eradication. And if immune escape prevails, if this wins, cancer mass can develop. If there is a imbalance between the tumor cells and the immune cells, if the tumors, the immune mechanism is more effective than the suppression, the tumors are eradicated and hemostasis should be maintained. And there are many actually immune escape mechanisms that involved with the immune suppression mechanisms. The tumor antigens are shed, and there is a cleavage of tumor antigens into the bloodstream. Anti-inflammatory cytokines are secreted, Sialoglycoprotein expression is important to hide the antigens. There is a diminished expression of tumor-associated antigens. And also, there are some caveats in anti-tumor immunity. Antigens are variable that are expressed on the tumor cell. There is a stimulation, co-stimulation defect. There is a suppression of antigen-presenting cells, especially CD1C positive cells. Cytokine synthesis is suppressed in many situations. And Treg stimulation is important. Treg cells are regulatory cells, are suppressor cells, and Th17 cells are actually anti-activated cells. They develop anti-tumor, anti-Th1 activity, and also Treg cells can help Th17 cells. And also class 1 antigen expression is decreased on many cells. With a couple of slides, I would like to show you the importance of angiogenesis, which is related to actually the anti-tumor immunity. Angiogenesis involves the sprouting of the vascular structures, because tumor cells need oxygen, and angiogenesis provides oxygen, the blood supply to the tumor cells. VAGF, vascular endothelial growth factor, is one of the most important factors that is related to the angiogenesis. Interestingly enough, it involves many characteristics of the tumor cells, including cell migration, proliferation, permeability, and VEGF is produced by many immune cells. So immune cells, when they are activated, secrete VEGF. It provides the vascular sprouting, vascular development, which is very important for the tumor mass proliferation. And also, it can cause proliferation and permeability. And also, these are very important in terms of metastasis. So these systems, the angiogenesis, the anti-tumor immunity, and the growth factors are interconnected by each other. But also, unfortunately, when the immune system is activated against tumor cells, these angiogenic factors are produced, and these factors help the tumor cells to proliferate, to prevent the immune activation, and also to do metastasis. There are many cytokines that are important for angiogenesis, but among them, interleukin 6 and interleukin 8 are most important ones, and they stimulate the tumor cells in an autocrine fashion. It means that there is a vicious cycle developed between the tumor cells and the immune cells, and this vicious cycle increases the vascularity more and more, and it cannot be sometimes suppressed by any molecule that are against angiogenesis. But the question is it the angiogenesis is important for lung tumors? It is very important, and there is a interface between the vascular system and the tumors. This interface is very important in terms of vascularity, the angiogenesis. Inside the tumor, there is a hollow core full of necrotic cells, and the vascular sprouting is not effective for these cells because they are all mostly necrotic, but the growth components actually are here just at the interface of this cell, of this two compartments. And in this compartment, VEGF is secreted by the tumor cells as well as the immune cells. There are many pro-angiogenic endogenous molecules, and also for inhibition of these angiogenesis, there are a lot of anti-angiogenic factors, endogenous factors. In summary, there are many cells that are active, that attack the tumor cells, such as natural killer cells, macrophages, T lymphocytes, but also there are other cells that are by definition should by definition attack the tumor cells, but become a helper of the tumor cells. They produce suppression to suppress the anti-tumor cells, T lymphocytes, natural killer cells, and also they provide more angiogenesis for the tumor cells, and also they help the tumor cells to metastasize more easily. So there is a equilibrium mechanism called immune surveillance. The immune cells actually are the surveillance cells to find and kill the tumor cells, but because of the anti-tumor, anti-inflammatory mechanisms, the suppressive mechanisms, this cannot happen in some of the situations, and this is why we see clinically evident tumors in 1/3 of whole population. Of course, there have been many immunological therapies that are promising. Some of the anti-tumor immunological therapies have been accepted to be used in humans, but actually a very small fraction of immunotherapies have been found to be effective against tumors. One of the major reasons for that, the immune system are more complex than it was thought to be, and this complex system should be considered in terms of the development of new immunotherapeutical drugs or procedures. But probably in future, we will be seeing more and more immunologically active therapeutical options. Thank you for your attention.
Excellent, excellent. This is by far the best immunology lecture I've ever heard in thoracic surgery. I have to say, it is a very, very, very complex topic, but you have covered it very well. And I'm going to ask you a few questions to simplify, because I'm pretty certain our audience is completely, confused now. Because yeah, they're lost. They've lost hope in immunology. So we will try and simplify the topic. It's just amazing. Amazing what you just presented, and your slides are just A1. I'm so happy to have this lecture in the collection, because I think we'll have to keep going back and listening to this talk again and again, particularly looking at your slides to understand, what is happening. So let's start with some question and answers. Is that okay?
Yes, sure.
Akif, yeah. So stop sharing your screen, and then we'll start with the first thing. So I'm going to just come back with first, before going into the very basics, I'll ask you a simple question. You said that our body is producing every day at least 4 to 5 cells which could be immunogenic, right?
Yes.
Yeah. Stop sharing your screen so that we can see you. Okay?
Sure.
Yeah. Okay.
Yeah.
Yeah. So stop the screen share. We are still sharing your screen. So I'll just stop your screen sharing. So now you can come on.
Sure.
And I'll put you on. Okay, fantastic.
Okay.
So you said that our body is consistently producing 4 cancer cells which have, not cancer cells, but cells which have a potential to develop cancer every day, about 5 to 6 cells. And then we have a response by the body, either with the, can others switch off your microphones? Because there is, sorry, just one minute. Yeah, okay. So you said that our body has a response from either by the T lymphocytes or by the natural killer cells. And they try to take away the, they try to kill the potential cancer cells. So this concept of immune surveillance, how can we put it into clinical practice, for surgeons to understand how can we do an immune surveillance to know that there is a potential of developing a cancer in future? So what is the mechanism of doing clinical immune surveillance of a normal individual?
Okay, thank you very much for this question. Actually, there are some molecules, there are some compounds that have been shown to be activator of immune cells. And some of the molecules are very important in terms of developing normal anti-tumor activity. In 1980s, vitamin C and some other molecules were thought to be very important for immune system. But none of them were proven to be effective. Today, as a short answer to your question, in order to increase our immunity, we have to be very careful about vitamin D level. Of course, there are other factors that are important for normal immune system, because of course it should be a good idea, or it could be thought as a good idea to increase the immune system in order to increase the effectiveness of the immune surveillance. But when you increase the activity of immune system, for example, adjuvants, some of the adjuvants which is found in the BCG vaccination is very effective in order to increase the immune system. But you can cause autoimmune phenomena, autoimmune diseases, and you can cause some of the derangement of the immune system. So without doing anything, it has been shown that only vitamin D is important in order to keep your immune system very effective. There are two other things that have been shown to be effective for the induction of immune system. It is also these two things are important in terms of pandemic. One of them is good sleep. There are some mechanisms involved with this. Probably melatonin is one of them. The other one is exercise, regular, middle intense exercise. So we should increase our anti-tumor immune system with three things. Actually, I advise my patients after oncological surgery to take some vitamin D if it is lower than the normal, and exercise and sleeping. So there are many possible and proposed molecules, some drugs that are thought to be important in order to induce the immune system, but none of them was proven to be effective in terms of tumor immune surveillance against tumors.
But do we have any particular, particularly in terms of lung cancer, do we have any immunological agent that we can look at which will tell us that you are developing a lung cancer? Is there any marker, immunological marker in the blood at the moment?
No. The problem is that there are many immunological markers, because as I showed you, interleukin 6, 8, 2, 10, TGF-beta are important, but you can find these immunological markers in normal situations. Interestingly enough, for example, some molecular studies, proteomics studies, researchers found many proteins that are shown to be involved with the tumor development. But most of them are immunological factors, not tumor-specific factors. So many immunological factors are there, but none of them are specific for tumor. I mean, the expressions are higher, but the expressions are higher in the simple flu. It is higher than in the state of the chronic bronchitis. It is normal sometimes because of the very simple infection, such as urinary infection. So unfortunately, none of them have been found to be specific for tumor development. So until now, there is no immune parameter that can be used for tumor detection.
Okay, so we cannot use any immune marker for screening of lung cancer at the moment.
No, unfortunately, no.
Okay, that's one. Now, the other thing which I just want to tell the audience, because they are very young surgeons there, is that the two concepts, one is apoptosis, which is programmed cell death, and that happens either because of gene amplification, gene deletion, gene rearrangement, and gene mutation. These are the four mechanisms by which programmed cell death happens. And a disruption in any one of these four mechanisms is what causes cancer. Is that correct?
Yes. But in some tumors, all four mechanisms are disrupted, not one or two.
Okay. And so when we give immunotherapy in a patient with lung cancer, which one of these four are we trying to do? What do those immunotherapy agents do?
Actually, now there are many immunotherapeutical options for some tumor cells, but very small number of them are effective and are clinically used. For example, take PD-L1. PD-L1 is so important that the two physicians received a Nobel Prize for this discovery last year in 2019. PD-L1 is there. Remember the graphs, the mechanisms. PD-L1 is there in order to suppress the tumor, anti-tumor T lymphocytes, and also natural killer cells. It works as a suppressor. Also, if the bounding time is higher enough, because there is an activation threshold dependent on the binding time and the energy, the thermodynamic energy between the ligand and the protein. If it binds long enough, it induces T cell apoptosis. So anti-PD-L1 therapy, for example, pembrolizumab, inhibits this inhibition because tumor cells highly expresses this PD-L1 on the surface, and these PD-L1s should be there, but they should be expressed in a very small amount because it shouldn't, the anti-tumor immunity shouldn't be suppressed. But if we give anti-PD-L1 antibody, we can suppress this inhibition. And this inhibition, this suppression of inhibition, it is like cutting the brake of the immune system, because it works as a brake of immune system. This brake is used by tumor. When we cut this brake, the immune activation is increased. And this is why we saw this therapy is so effective against lung cancers. There have been many studies. For example, in 1990s, I started to work on tumor immunology in 1988 when I was a second-year medical student. In those times, we thought that there would be a tumor vaccine, because vaccines are so effective against virals and the bacteria. And tumor vaccines would be effective against tumor. There have been many vaccines developed during this this 3 decades, but none of them was found to be effective against tumor. Because with vaccine, you can develop anti-tumor immunity, but there is a break against anti-tumor immunity. There is a very good suppression against anti-tumor immunity led by tumor cells, macrophages, Th17 cells, Treg cells, and neutrophils. So today we have seen that anti-PD-L1 therapy is very effective. There are some effective therapeutical options in, for example, lymphoma. Anti-CD20 antibody is effective in some of the lymphomas that express CD17. There are some other immunological therapies, but anti-PD-L1 therapy is most effective one to date.
Okay, so pembrolizumab is used in lung cancer. So when we send the tissue away to the pathologist, do they look for the PD-L1 ligand in the tissue, or is it available in the blood sample? Where do they look for the PD-L1?
Okay. If you look into the bloodstream, actually you can find many PD-L1 components in us, actually, you and me, normal people, because PD-L1 is there for normal suppression. Every day, billions of cells, T lymphocytes, are suppressed because they tend to attack our cells. And there is a suppression system to block these cells to attack our cells. So PD-L1 is normally there. And the level of PD-L1 is not high. This is why the pathologists look at the PD-L1 expression onto the tumor cells. But there is a problem with that. Normally, if the expression rate is higher than 1%, there is a good possibility that anti-PD-L1 therapy could work. But if it is higher than 50%, the therapeutic action should be higher than expected, or very high. But the situation is not like that in some patients, because the tumor cells, as I showed you, tumor cells are very heterogeneous. Pathologists cannot look at whole billions of cells. Some of them are expressing PD-L1, but some of them are not. This is why anti-PD-L1 therapy is given to the patients in which the tumors are expressing PD-L1 more than 50%, but it is effective in the patients with tumor expressing less than 1%, because some other tumor cells can express PD-L1, and also anti-PD-L1 therapy is effective against the tumor suppression led by normal cells. So anti-PD-L1 is effective by many mechanisms. This is why PD-L1 is important in terms of the prediction of the anti-tumor immunity or the effectiveness, but sometimes it doesn't say anything about the effectiveness.
Yeah, so no, my question was, clinically, what do we do when we send the specimen and we ask for PD-L1? Is it because we are trying to decide whether this patient will benefit from immunotherapy or not?
Yes.
Is that why we are doing PD-L1? Okay, and the immunotherapy, that if the expression comes more than 50%, then we consider them for pembrolizumab. Is that the clinical implication, right?
Yes.
I'm trying to simplify it for the juniors, just so that they understand. So there is some role of immunotherapy in lung cancer, depending upon PD-L1 expression. And that is what you ask for when you send the tissue away for analysis, is PD-L1 ligand. Okay, so that is just, I'm trying to simplify it for the juniors so that they understand how to put this knowledge into clinical practice. Now, the other thing which these guys need to know, because these are thoracic surgeons, not oncologists, they need to know four receptors: KRAS, BRAF, BRAF, HER2, HER2, and P53. Is that correct?
Yes.
These are the four important ones in lung cancer. And when we do a immunohistochemistry, this is what they look for. Is that correct, Akif, or is it something different?
KRAS is important. Also, KRAS is important. Yeah.
So KRAS, BRAF, HER2, and P53. When you go into the exams, if you are ever asked about oncogenes and things like that, these four names have to come out from the tip of your tongue. Okay, it's important. And
also EGFR is important.
Yeah, and EGFR. Sorry, I forgot that. Yeah, I forgot EGFR as well. So EGFR, KRAS, BRAF, HER2, and P53, 5 of them. Okay, and you must know a little bit about each one of them. So please go back to his slides. He presented beautifully about each one of these: EGFR, KRAS, BRAF, HER2, and P53. Go back. I think it was the third or fourth slide. I would recommend everybody go back to that and try to understand what he was talking about in terms of these 5 markers. Because this is what you will be asked in the exam. You will be asked about these 5 markers if the discussion goes down to immunohistochemistry. P53 is specific for squamous cell carcinoma. Is that correct?
Actually, it is not so specific because it can be found in almost all lung cancers. But in small cells, it can be found 3/4 of the cells. And in squamous cell and the adenocarcinoma, roughly 50 to 60% of the cells can express P53. So then we call P53, we are talking about mutated form of TP53, because TP53 is the wild type anti-oncogene, which should be there in all cells. Normally, we have TP53 wild type, but when we call P53, we are talking about the mutated TP53.
So now my question to you is, how does chemotherapy affect these immune markers?
Actually, it varies, because the chemotherapy, in essence, basically kill high proliferating cells. This is why lymphopenia or cytopenia develops actually in all patients who are receiving chemotherapy. So it kills almost most of the cells, not most of the cells that are effective to tumor cells. This is why actually during chemotherapy, the organism becomes more vulnerable to tumor cells. And also this is why immunotherapy plus chemotherapy at the same time, consecutively, is not a good idea to give any cell. But in some chemotherapeutical agents kill some of the suppressive cells more selectively, but it varies from one patient to other patient. So we cannot define the deleted or killed cells by chemotherapy specifically. But it is fair to say that it creates a very extensive suppression against tumor cells. So of course, it actually creates a immunosuppressive situation against the bacteria. This is why some of the patients can develop febrile neutropenia. They can develop septic conditions.
Sepsis, yeah. Okay. So the other interesting thing which you said, in fact, you started your lecture beautifully by saying that cancer or tumor is actually a monoclonal cell. It starts as
Yes.
one cell.
One cell.
But tumor is heterogeneous, which means that there are many different differentiation of the cells within the tumor. And the other very interesting thing which you pointed out very clearly for the audience was that metastasis of a tumor has two bases. One is an immune-based mechanism, which actually promotes metastasis. And the second one is an angiogenic mechanism, which also promotes metastasis. Is that correct? Is my understanding correct?
Yes.
Okay, and then
Also,
when we
yeah.
Yeah, and when we try to base immunotherapy, we try to base it either to attack the immune-based mechanism or to attack the angiogenic mechanism. Is that correct,
Yes.
Akif?
Yes. But if we look at anti-PD-L1 mechanism, there is no so far evidence that suggests that the anti-tumor agents can block also angiogenesis. But in theory, it should block angiogenesis. But there are some anti-angiogenesis agents, such as bevacizumab, in order to block angiogenesis. And there have been some studies going on with anti-angiogenic factors and also immunogenic factor, anti-immunotherapeutical agents. My immunological PhD, the thesis of my PhD was about investigation of the effectiveness of interleukin-12 and also anti-angiogenic factor in order to understand to block angiogenesis and the effectiveness of anti-angiogenesis and the immune stimulation together against tumor cells.
Okay. Just for the audience, could you just name it, no need to give details, but just name a few important studies which they can reproduce in the exam in terms of immunotherapy of lung cancer. If you know of any few important studies which they should know.
Actually, Pacific trial is important. Durvalumab, actually, it is a game-changing trial from my perspective. Pacific trial is a trial that showed that anti-PD-L1 therapy is effective against tumors, which is very, very important. And there is a keynote study. Keynote study again is a study that shows the effectiveness of anti-PD-L1 study. But the most important one is Pacific trial, I would say. There are many other trials.
Yeah, sure.
But the most important trial is Pacific trial.
So in the exam, if they just mention Pacific trial and rattle it out a little bit, they should look impressive.
Yeah.
So now I'm going to take a few questions from the audience. Shubham Garg, who's a surgical oncologist and obviously deals a lot with oncology, is asking us a few questions. So when we test for PD-L1 in lung cancer, at which level of immunity are we testing?
Which immunity? Actually, we are testing the anti-tumor immunity led by T lymphocytes. So since PD-L1 is a good suppressor molecule, it's an important suppressor molecule for T lymphocyte activity, it actually, when you give anti-PD-L1 therapy, you block the blockage of the anti-tumor T lymphocyte response. But PD-L1 is important in terms of anti-tumor immunity driven by macrophages also, and also neutrophils. But T lymphocytes are the most important one. So we would say that PD-L1 is a PD-L1 level is a good indication for anti suppressive anti-tumor suppression activity. So it is a marker of the suppression of the anti-tumor activity.
Okay, that's good. This next question
it is not a direct activity, it's a suppression, suppressive
so tumor
activity.
suppression is what you're looking at when you're looking at
Yes.
PD-L1.
Yes.
So Shubham continues to ask, saying PD-L1 testing is dependent on the type of platform that we use. Ventana versus DACO, which use different antigens. Is it possible that PD-L1 on Ventana comes negative, but that on DACO comes positive? So should we test on both platforms?
Okay, so it is a question for pathologists, but I know this discussion, and actually, you know, they test the PD-L1 frequency, the activity or the expression by a part of the peptide, the antigen. So they don't test whole antigen. This is why these platforms detect different types of different side of the antigen. So Ventana is the most effective one as far as I know. It is possible to, of course, to test the anti-PD-L1 activity by Ventana platform, and also you can switch to the DACO platform. But usually, pathologists, because of the financial reasons, pathologists choose one platform or other platform. They don't do two tests. It's a money-consuming thing. But in theory, yes. But there is one problem that involves with this situation that I mentioned before, that tumor heterogeneity is so high that you have to test a sufficient number of cells in order to get a real idea, real number of the cells that express the PD-L1 protein. So the heterogeneity is bigger problem. So you have to use one platform, which usually Ventana platform, they say is the best. But of course you can use the two platforms, but you have to have enough tissue.
Okay, does the decision to test for PD-L1, does it depend on the stage of the lung cancer, or should you do it for all lung cancers? Like if it's a T1 or a GGO, less than 1 centimeter, should you bother doing PD-L1? Or should you keep PD-L1 for the 2As, 2Bs, 3As?
Yeah, because of the results of many studies that are published, and there are a number of studies that are going on, pathologists is becoming to say that we should look at PD-L1 in all patients. Okay. All patients who are operable, who have been operated, who had operation, and all patients who are not operable, the patients with locally advanced tumors. Why? Because in locally advanced tumors, there is a good option, anti-PD-L1 therapy or anti-CTLA4. CTLA4, I showed you the CTLA4, but I didn't mention too much. Anti-CTLA4 antibody which is ipilimumab, is effective again, is important. It is effective for inoperable patients. Also, probably it is effective as an adjuvant therapy. In coming years, probably we will be using these immunotherapeutical agents as one part of adjuvant therapy. So this is why we should look at the anti-PD-L1 in all patients. Also, if these patients develop metastasis, which unfortunately 50% of all operated patients develop eventually a metastasis, we should have some idea about the PD-L1 expression in these tumors. So the oncologist can use immunotherapy in these patients.
So in a stage 1A, 1B, where we are not going to give any adjuvant therapy, you still advise to do PD-L1 so that in future, if they develop any metastases, you will have the information and then you can start immunotherapy. Is that correct?
Yeah, because
what you just said?
Yes, because in stage 1 patients, 1A, 1B patients, unfortunately develop metastasis. They do. 1/5 of them develop metastasis because 5-year survival is about 80%. So if the PD-L1 is known, an immunotherapy can be given. Actually, we performed operation to the inoperable patients, in 3 inoperable patients, in order to take biopsy from the inoperable tumor. Actually, we will see probably more and more indications like this, because oncologists want to know if the recurrent tumor is expressing PD-L1 or not. So there is another situation, because even the fact that PD-L1 is low or high in the primary tumor, the metastatic tumor can express different level of PD-L1 or CTLA4.
Okay. The next question is asked, what are the prognostic and predictive significance of the biomarkers that we mentioned? EGFR, KRAS, BRAF, HER2, P53.
Yeah. EGFR, actually, for EGFR, the studies are not consistent. The Asian patients, especially the patients in China and in Japan, EGFR is a bad prognostic factor for the patients. Actually, we looked at the EGFR expression in order to say if it is a factor of the prognostic factor. We didn't find any effectiveness or the importance of these factors. But other immunological factors, we don't know. There are some studies showing that PD-L1 is one of the most important prognostic factors. Actually, we have been conducting 2 studies right now in order to show if PD-L1 expression on natural killer cells and T lymphocytes are prognostic or not. I think this is more important than the expression on tumor cells, because the expression is variable on the tumor cells, but probably the PD-L1 expression on immune cells can be prognostic factors in terms of survival or recurrence. What we don't know. The short answer is no. There is no known prognostic significance until now. In 8th, 9th staging system, we will be looking at these factors, because all immunological and the molecular factors can be entered into the system, 9th staging international staging system database. But the problem is very few centers, very few institutions look at these whole factors routinely. But probably we will have a good information in terms of prognostic importance of these factors.
Okay. Two more questions to go. One is PD-L1 expression. Does it depend on the histology of the lung cancer?
Yes, on squamous cell cancer tumors, it is more and more the frequency is higher than the adenocarcinoma. But on some of the adenocarcinoma cells, it can be found, it can be expressed, and also small cell tumors are important. At the beginning of these trials, PD-L1 was assessed, was looked at in only patients with squamous cell carcinoma. But today, we know that it is effective against other histologies. When if there is a higher level of PD-L1 expression.
Okay. And the other question was that, chemotherapy we give for performance status 1 and 2 patients because it's a more generic treatment. Immunotherapy is more targeted. So does the performance status of the patient matter when you're giving immunotherapy?
Actually, it's a question for my wife because my wife is a medical oncologist. But as far as I know, they administer these immunotherapeutical agents to almost everyone. Actually, when usually they tend to make a decision like this, if the patient's performance status is 3, sometimes if it is not good, even the fact that the patient is a good candidate for chemotherapy because of the stage and other parameters, they don't want to give chemotherapy, but they give immunotherapy because the older patients, the patients with bad performance status can tolerate, as far as I know, yes, immunotherapy. But there is a problem with that because in some patients, autoimmune reactions, autoimmune syndromes can develop because of the immunotherapeutical option, immunotherapy. So one should be very careful about this unexpected reactions, such as thyroiditis, pneumonitis, not pneumonia, but they can develop pneumonitis. Some immune reactions against adrenal gland or some other glands. Sometimes pancreatitis and other problems can develop in these patients.
So the reality is, Ramaswamy, immunotherapy is definitely better tolerated by yes, patients. They definitely have less side effects as compared to chemotherapy. Chemotherapy generally has yes, a lot more side effects. But when yes, the side effects of immunotherapy kick in, they can be pretty serious because autoimmune response is a big issue. And these patients can become ill very fast. So you cannot give yes, immunotherapy willy-nilly to everybody. You still need to make sure that a patient can afford it, and B, most importantly, it's very expensive. Okay, it's a very expensive treatment at the current moment. I think immunotherapy is only to be used within the domains of trials rather than generic use. I think patients, if they are going to receive immunotherapy, they should be enlisted within a trial. That is what is the recommendation. At least in the UK, we don't give immunotherapy randomly. We would give it only when they are part of a trial or part of a study. Because this is an NHS, we've got to ration the treatment. And so it is quite an expensive business, actually. It's not, these are new drugs. So just one more point which I want to make for the exam going people. I would suggest you go back to Professor Turna's slides and look at a couple of slides. One slide where he spoke about natural killer cells. I think it's very important. You must know a little bit about natural killer cells and the various actions that they have. And the other one that you must know, and he's got a beautiful slide on it, is on T lymphocytes. Particularly, you must know the Th1, Th2, and Th17. Okay, so you got to look for cellular immunity, humoral immunity, and tissue inflammation. He's got a very nice slide where he made a table and he explained Th1, Th2, Th17. Okay, I suggest you go back and look at these slides again. Thank you very much, Professor Turna, Akif, my dear friend. It's just such a pleasure to listen to you. And I think you are one of the few surgeons in the world who understand immunology so well. You get so excited by immunology. I can see that you know it inside out. So you're just the perfect guy to give us this talk. And honestly, I learned a lot today, and I'm sure my students have learned a lot. They must be a little confused, but that's all right. It's better to know what you don't know. And so you can then go back and address it. That's why we are recording these talks, You will not understand it in one go. I promise you that. There were two talks. One was this tumor immunology. And in my lifetime, I'm telling you, one was this tumor immunology. I've heard this in the past. And the second one was precision medicine. These were the two talks in which for 2.5 hours, I didn't understand one word on the slides till I went back and read these slides again. Okay, so it's not an easy topic, but you have the right man to guide you. And he has put it all in pictures. That is the beauty of his slides. Everything is in pictures. So please go back and listen to this talk again and again. It'll take you a little time, but this is the future. This is, if you are ahead of the game and you understand that immunology is the future and not surgery, then probably 10 years down the road, you will be richer than what you are today. Okay.
Yes.
Thank you very much. Thank you,
Thank
Akif.
you.
It's really been a pleasure talking to you. And thank you everybody else for logging in. We're going to actually log out of this lecture and then we will log back in at 5 o'clock. Is that okay? Because we've got another lecture on tracheal surgery from Dr.
Thank you
Santosh.
very much for your tolerance to me, because
No, it's brilliant.
You could develop anti-Turna immunity, but you didn't.
No, it's brilliant. I think your knowledge of immunology is just, I bow to your knowledge.